Hello everyone. In this video, we discuss about glycolysis process. First of all, what is mean glycolysis? Glycose means sugar. Lysis means breaking down. Breaking down of the glucose, the pathway that is converted into glucose is converted into pyruvic acid in several steps, and that's why this name it as a glycolysis process. Here, this pathway, in other words, it is also called as a EMP pathway. Why it is referred as a EMP pathway? Because there are three scientists. They have worked on this particular EMP, this glycolysis process. One is Embeden, Gustav Embeden, and Otto Meyerholf, and parnas. There are the three scientists. They have worked on this particular glycolysis process. That's why it is referred to as EMP pathway. The starting their names, respective of their names, they can able to call it as the EMP pathway. Embeden, Meyerholf, and parnas. Three scientists they have worked on this particular pathway. That's why it is named as EMP pathway. This is the most common pathway that takes place in all living organisms. And here in the anaerobic organisms, this is a process. Only this is the process. The respiration takes place, and that's why this is the most essential pathway that is occurs in the anaerobic organisms, and also takes place in all living organisms. Most of the all living organisms, from the plants to animals, most of the living organisms, this process takes place. And this process that occurs in the cytosol of the living organisms, cytoplasm, takes place in the cytoplasm. And also in plants, if you observe in the plants, sugar molecules that are produced by the process of the photosynthesis process. Okay, here in the photosynthesis process, it produces sucrose molecules. Sucrose molecules are produced. Now the sucrose molecules now converted into glucose or fructose with help of the enzyme that is invertase enzyme. The invertase enzyme that convert the sucrose into glucose or fructose. And further this enter into the glycolytic pathway and starts glycolysis pathway. Here, first of all, let me discuss about each and every steps of this particular glycolysis pathway. Here, glycolysis that occurs mainly in the three distinctive phases. One is energy consuming phase, and other one is spreading phase, and other one is the energy producing phase. These are the three different steps that occurs here. First, glucose. Glucose is converted into, when glucose enter into the cytoplasm, the phosphorus molecules that comes and attached to this particular glucose molecules, it is also called as phosphorylated, and the glucose is phosphorylated and formed into glucose 6-phosphate is formed here with help of the enzyme that is called hexokinase enzyme. Hexokinase enzyme that converts the glucose into glucose 6-phosphate. Here 6 molecules of the glucose is converted into glucose 6-phosphate. Here when hexokinase enzyme is acted upon this, and here now is here also ATP consumed here. ATP is converted into ADP here. Adenosine triphosphate that converted into adenosine diphosphate that is formed here. Now formed glucose 6-phosphate, now that is converted into fructose 6-phosphate. Here enzyme involved here, hexose phosphoisomerase enzyme. Hexose phosphoisomerase enzyme that convert the glucose 6-phosphate and into fructose 6-phosphate. These glucose 6-phosphate and fructose 6-phosphate are the isomers. These isomers now that convert this particular converted by the enzyme hexose phosphoisomerase enzyme that will convert this particular glucose 6-phosphate into fructose 6-phosphate. Now formed fructose 6-phosphate Now that is converted into fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate is formed here. The enzyme involved here, phosphofructokinase enzyme. This step also energy consuming process. ATP is converted into ADP here. And these are the two steps, that is energy consuming process, means where that energy is utilized for the formation of the phosphorus molecules. Now the formed fructose 1,6-bisphosphate, now it is splitting into, now this step that is called splitting phase. Now fructose 1,6-bisphosphate, now that is converted into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, DHAP, and glyceraldehyde 3-phosphate. These two molecules are triose phosphate molecules, means here the 6 molecules of fructose 1,6-bisphosphate, now that is converted into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. These two molecules are interconvertible, means glyceraldehyde 3-phosphate may be converted into dihydroxyacetone phosphate. Dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate. Depends upon the requirement of the cell, that is interconvertible. Here further this DHAP, that is dihydroxyacetone phosphate is converted into, that is entered into the different pathways. Here aldolase, the enzyme involved here, aldolase enzyme, and here that is interconvertible enzyme is called triphosphate isomerase enzyme. The triphosphate isomerase enzyme that convert the glyceraldehyde 3-phosphate into dihydroxyacetone phosphate or dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. These are the interconvertible compounds. Now formed glyceraldehyde 3-phosphate, now that is converted into 1,3-bisphosphoglyceric acid is formed here. Now it is converted here. Now enzyme involved here, glyceraldehyde 3-phosphate dehydrogenase enzyme involved here. Glyceraldehyde 3-phosphate dehydrogenase enzyme, now that is converted into glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. Here 1,3-bisphosphoglyceric acid, now formed 1,3-bisphosphoglyceric acid, now that converted into 3-phosphoglyceric acid. 3-phosphoglyceric acid is formed here. Here also in the above step also energy producing phase, means NAD is converted into NADH and H plus is formed here. Nicotinamide adenosine dinucleotide, that is converted into nicotinamide dinucleotide, two hydrogen molecules. 2 hydrogen molecules are formed here. Here the triose bisphosphate, means 1,3-bisphosphoglyceric acid, now that is converted into 3-phosphoglyceric acid is formed here. Triose phosphate. Now enzyme involved here, phosphoglycerokinase enzyme. Phosphoglycerokinase enzyme, then now that is converted into 1,3-bisphosphoglyceric acid into 3-phosphoglyceric acid. Now here enzyme involved here, phosphoglyceric acid. Now here also ADP is converted into ATP. Adenosine diphosphate is converted into ATP, means energy producing, means energy is produced here. From here onwards, that is energy producing here. Now formed 1,3-bisphosphoglyceric acid is converted into 3-phosphoglyceric acid. Here ADP is converted into ATP, 3-phosphoglycerokinase. Now 3-phosphoglyceric acid, now that is converted into 2-phosphoglyceric acid. 3-phosphoglyceric acid converted into 2-phosphoglyceric acid. In the enzyme involved here, phosphoglyceromutase enzyme. Phosphoglyceromutase enzyme, now that is convert the 3-phosphoglyceric acid into 2-phosphoglyceric acid. Now 2-phosphoglyceric acid, that is converted into here removal of the water molecule takes place. Water molecule comes outside, and so that the 2-phosphoglyceric acid is converted into 2 molecules of the phosphoenol pyruvate is formed here. The enzyme involved here, enolase enzyme. Enzyme involved, enolase enzyme. Now that is the 2-phosphoglyceric acid converted into 2 molecules of phosphoenol pyruvate is formed here. Now formed phosphoenol pyruvate, now that is converted into pyruvic acid. Pyruvic acid formed here. Here also ADP is converted into ATP. Enzyme is produced here. Pyruvate kinase enzyme. In the presence of the enzyme, pyruvate kinase enzyme that convert the 2-phosphoenol pyruvate into pyruvic acid. These are the steps that involved in the glycolysis process to convert the glucose molecule into pyruvic acid. Now let us discuss each and every step here again. The glucose is converted to glucose 6-phosphate first, and first later the glucose 6-phosphate is converted into fructose 6-phosphate. Fructose 6-phosphate is converted into fructose 1,6-bisphosphate. The formed fructose 1,6-bisphosphate now splitting into glyceraldehyde 3-phosphate and dihydroxyestone phosphate. Now the formed glyceraldehyde 3-phosphate now that is converted into 1,3-bisphosphoglyceric acid. Now the formed 1,3-bisphosphoglyceric acid now converted into 3-phosphoglyceric acid. These are the triose phosphate molecule, 3-phosphoglyceric acid. Now 3-phosphoglyceric acid now that is converted into 2-phosphoglyceric acid. 2-phosphoglyceric acid is formed here. The formed 2-phosphoglyceric acid that is converted into two molecules of the phosphoenol pyruvate is formed here. The formed phosphoenol pyruvate now that is converted into pyruvic acid. These are the molecules that is formed during the glycolysis process. Enzymes involved here, first one is hexokinase enzyme. Here also ATP is converted into ADP, and glucose 6-phosphate is converted into fructose 6-phosphate. Here enzyme involved here, hexose phosphoisomerase enzyme. Means these are the two molecules are isomers, so that isomerase enzyme is involved here. Now fructose 6-phosphate is converted into fructose 1,6-bisphosphate. Here also energy consuming process, and the phosphofructokinase enzyme that is involved here. ATP is converted to ADP. Fructose 1,6-bisphosphate. Now the formed fructose 1,6-bisphosphate now that is splitting into glyceraldehyde 3-phosphate and dihydroxyestone phosphate. Triose phosphate and triose phosphate. Two molecules of the triose phosphate is formed here. Now the glyceraldehyde 3-phosphate now that is converted into 1,3-bisphosphoglyceric acid. Here that is enzyme involved here, glyceraldehyde 3-phosphate dehydrogenase enzyme that is involved here. Glyceraldehyde 3-phosphate dehydrogenase enzyme now that is converted into glyceraldehyde 3-phosphate into 1,3-bisphosphoglyceric acid is formed here. Now 1,3-bisphosphoglyceric acid now that is converted into 3-phosphoglyceric acid. The enzyme involved here, triose phosphate. Here phosphoglycerokinase enzyme involved here. Now ADP is converted into ATP here. Now formed 3-phosphoglyceric acid is converted into 2-phosphoglyceric acid. Enzyme involved here, phosphoglyceromutase enzyme. Means the 3-phosphoglyceric acid is converted into 2-phosphoglyceric acid in the presence of the enzyme phosphoglyceromutase enzyme. Now here afterwards the 2-phosphoglyceric acid now the water molecule that comes outside and formed into two molecules of the phosphoenol pyruvate is formed here. Phosphoenol pyruvate is formed here. Now phosphoenol pyruvate now that is converted into here the enzyme involved here, enolase enzyme. The phosphoenol pyruvate that is converted into pyruvic acid, the pyruvate kinase enzyme. Here also ADP is converted into ATP. Here this is the step that is produce the energy from the glucose molecules here. Here the three steps are involved here. One is NAD is converted into NADH and H plus, and this step is also produce the ATP. ADP is converted to ATP during the 1,3-bisphosphoglyceric acid and 3-phosphoglyceric acid. 1,3-bisphosphoglyceric acid and 3-phosphoglyceric acid. Here also ADP is converted into ATP. And the final step, the phosphoenol pyruvate is converted to pyruvic acid. Here also ADP is converted to ATP molecule. These are the three steps that is produce the ATP molecules. Here one NAD is equals to 3 ATP molecules. 3 ATP molecules are produced. So that NAD is converted to NADH and H plus. And here 3 molecules of the ATPs that is produced here. And this is about glycolysis pathway to convert this particular glucose into pyruvic acid with the help of the various kind of the steps that are involved here, and produce the energy and consuming the energy and forms into pyruvic acid. Thank you everyone, and please watch and subscribe my channel. Thank you very much.